Hello, everyone. It's my pleasure to introduce the speaker for today, Dr. Ding Zhu from University of Colorado Boulder. So Ding has been here now on campus, or at the Boulder campus for 21 years. In fact, he started right when I started, so in 1997. And Ding had done his graduate work with Martin Chalfie at Columbia University. This was before Martin Chalfie got his Nobel Prize. And then he went on to do postdoctoral work, also in C. elegans, with Bob Horowitz at MIT. And again, this was before Bob Horowitz got his Nobel Prize. So I think the lesson here is whoever's lab Ding goes and works in goes on to get the Nobel Prize. So I'd like to formally invite Ding to do a visiting professorship in my lab. But he did really fundamental work in Bob's lab, Bob Horowitz's lab, on understanding the mechanisms of apoptosis. And this is before a lot of these pathways became well known within other systems. And a lot of the basic fundamentals are worked out in the C. elegans system. And as we've seen, that, this is a great example of how a very basic model system can lead to huge implications in not only all other organisms, but have big medical implications. There are a large number of drugs now targeting pathway components in the apoptotic pathways. So in his own lab, he's gone on to really go from A to Z in understanding apoptosis, how the cells are actually recognized, how they're engulfed, how they degrade the DNA. And some of this has led to new directions in the lab, such as understanding how it is that paternal mitochondria are eliminated from, during sexual reproduction. So basically, they're marked and eliminated by a mitophagy-mediated pathway, and his lab worked that out. He's also followed other directions, now understanding hepatitis, studying hepatitis B, and understanding the role of this X protein in that. And finally, his most recent forays have led him into understanding bystander effects with radiation. And that's what he'll talk about today. So you'll see that, again, this basic understanding that you can get from using a model system like in C. elegans can really lead to new fundamental understanding of pathways that have clear relevance in human disease and in human medical treatments. But one thing I'll mention before we welcome Ding, we're going to have the usual Cancer Center little short talk from one of our shared resources. And today we have the pleasure of having Karen Helm, who has been, I think, running the flow cytometry shared resource, since I think the early 1900s, right? And so she has been a real asset to the University of Colorado campus in her knowledge and understanding of flow cytometry. So she's going to give a few minutes introduction to talk about some of the latest capabilities of this shared resource. And then we will welcome Ding. Thanks.
Thanks, James. So, this is going to be tricky. His PowerPoint's there and mine's there. Oh, thank you. This one? Okay, great. So I'm the manager of the flow cytometry shared resource, and I have been here almost 26 years. So I have a history here. We do a lot of different things. I'm sure that many of you are familiar with us, but some of our services are we sort cells quickly. We do multi-parameter analysis on analyzers, and that is has grown from initially doing one color when I first started in flow to 21, 22 different fluorochromes now, and that gets tricky, but we can help you with that. And we also have mass cytometry, which uses metal-tagged antibodies instead of fluorochrome-tagged antibodies. And by doing that, we reduce the amount of spillover between markers. And so instead of doing a maximum of, say, 20 or 21, we can go over 40. So I know several of you are using that. We have several projects going on, and we're more than happy to help you with that. And we have other things, Illuminex MagPix, which does basically microsphere assays in suspension and multiplexed, a cell counter, and we're more than happy to consult on any project you might dream up. So we currently have 4 cell sorters, And the Sony is brand new. And we also are about to get a SciTech Aurora, which I'll talk about. And then we have the mass cytometer. We have software licenses that you can either buy from us, or Kaluza is actually available at no charge. Oh, one other thing. Just so you know, there's an Amnis image stream, which is a combination of image analysis and flow cytometry available at the VA. There are a lot of people that have wanted this for a number of years, and so we're really happy that it's at the VA, because it's the kind of thing that really needs to be shared across multiple institutions. So the self-run Sony sorter is being installed as we speak. It's in the lab and being installed today. So it's a nice small sorter that's made to be a customer-operated sorter. So it has fairly intuitive software. But in addition, it's their newest in their line, and it has 4 lasers, which is great. That means it can do up to 12 colors and 2 scatter parameters, multiple different nozzle sizes, does 4-way sorting instead of just 2 like most self-run sorters do. And it just looks to be a really nice addition to the lab, especially for people that need to do after-hours sorting. We're going to have some training classes once we kind of learn it ourselves, and be offering training, and then you'll be able to probably operate it during the daytime for a few times when we're around in case there's problems, but then for those of you who really need nights and weekends, it'll be available. Our antibody bank that we have for our mass cytometry is merging with the HIMSR bank, so it's just going to be a lot easier to use. That's going to happen this week. It's going to have its own website and software, and we have about 400 antibodies for mass cytometry. And then coming very soon, and we hope that means within a month, we're getting a SciTech Aurora spectral analyzer. So a spectral analyzer analyzes the fingerprint, let's say, of each fluorescent dye and then unmixes them. So rather than having a dedicated detector for each color, it unmixes fluorescence across the spectrum, which makes it much easier to do really multicolor work. And I've heard from friends who have these that it's pretty easy to dream up a 20-color and not have to spend a lot of time at it, and it works. You can do combinations that you can't do on a normal analyzer. So those of you who are familiar with flow know that you can't normally do APC and Alexa 647 together, but you can do that on an Aurora. So I think it's going to be a really good addition. And here's our staff. We added a new person because we have such demand, especially for sorting. She's being trained now, and we have a total of 80 years of flow experience, so we probably know, or one of us has experience with almost any assay. So that's it.
Okay, so I want to thank James for inviting me to give the Cancer Center seminar for the kind introduction. And it's always good to be back to Anschutz campus, where I have a lot of good friends and colleagues. So as Jen mentioned, my lab actually work on multiple different projects, all health related. For example, that we have been working on apoptosis for in the last 20 years, studying the mechanism of apoptosis regulation, activation, and execution. We actually study mechanisms that establish and maintain phospholipid asymmetry. We work on mitochondrial inheritance and mitochondrial biology. We also use C. elegans as an animal model to study Hep B virus, how Hep B virus causes pathogenesis, and also screening for drugs that can treat Hep B virus cause liver disorders. So today I'm going to tell you a story that started as a very risky project that no postdoc or graduate student wanted to work on, and gradually evolved to become a major project in my lab. And that's radiation-induced side effect. So about 10 years ago, I was invited to a meeting, apoptosis meeting in Poland, and one of the keynote speakers, Kamil Mazurciel, actually is a major figure in the radiation-induced bystander effect field. And he actually gave a very interesting talk about radiation-induced bystander effect. You can see the title here. Are they destructive or protective? And Dr. Mazurciel also talked about the difficult challenge of identifying factors that are involved in radiation-induced bystander effect. So after the talk, I saw that maybe the C. elegans model can do something about it. And so just a quick introduction, because there's a big radiation oncology department here. So this shouldn't be a new topic. So radiation-induced bystander effect actually refers to the phenomenon that non-irradiated cells are affected by factors released from irradiated tissues or cells. And this is a topic very important to the side effect that it seems in human, for example, human cancer radiotherapy. And it's also a very important topic for radiation safety and radio protection. And as you probably all know, that cancer radiotherapy could be quite nasty because you can see all sorts of adverse side effects. It can induce, for example, skin irritation, fatigue, dry mouth, nausea, hair loss, low blood count, and infertility. And also, in some cases, these side effects can even promote cancer resistance to the radiotherapy. There is so far no good medicine to alleviate this radiotherapy-induced side effect. So as I mentioned, that I was wondering that whether we can use the C. elegans model to tackle this difficult question: is what are the factors that are involved in side effect, bystander effect? And to do this, we actually first have to know whether C. elegans actually has a similar bystander response. And we need an assay to identify Libby factors. Before I get into details, I want to just briefly mention the C. elegans model. You probably are pretty familiar with this because there are some really good C. elegans experts in this campus, like Tom Evans and the retired professor Tom Blumenthal, who is a very good friend of mine. So C. elegans basically is a very powerful animal model to study fundamental biological process. It's a small animal, only 1 milliliter long, with less than 1,000 cells. And it's transparent and has a short life cycle, only 2.5 days per cycle, per life cycle. That's really important, as I will mention, for our study. It has a completely defined cell lineage, so we can study a lot of biological process at the single cell resolution. It also has a very powerful reverse genetic tool, for example, RNAi, which was actually RNA interferon, which was invented in C. elegans, and the CRISPR-Cas9 mediated knock-in, knockout are right now very mature in C. elegans. So in the last 30 or 40 years, the radiation-induced bystander effect field had been using this medium transfer system to study bystander effect in the mammalian cell. So what the researcher did is to, for example, irradiate the culture human cells, and after a few hours, spun down the cells, take the supernatant, the cell-free supernatant, and then incubate the supernatant. They call this supernatant as irradiation conditional medium, or ICM. And incubate the medium, the conditional medium, with naive or unexposed cell, and then to analyze the bystander response at both the cellular and the molecular levels. We actually decided to adopt this approach to study C. elegans bystander effect. Okay, so what we did is very similar. We actually culture C. elegans animal in the liquid medium, irradiate them with UV or gamma irradiation, ionizing irradiation, and then we spin down the animals, take the supernatant, we also call it as a conditional medium, okay? And then we incubate the conditional medium with naive or unexposed worms. And then after 24 hours, we start analyzing what happened to these animals, whether they exhibit any similar response to what people think in the human radiotherapy. So, and the answer is yes, we actually see multiple bystander effect. For example, one is shown here that these conditional medium, after incubating with the naive worm, can actually significantly inhibit apoptosis in the C. elegans germ cells at a dose-dependent manner, which is another typical feature of radiation-induced bystander effect. We also see that these conditional medium can cause increased embryonic lethality, increased germ cell proliferation, lead to chromosome DNA damage in the germ cells. I'll show some example later on. It also induces stress response, which is also, or some of this phenotype that we see are all kind of prominent symptoms of radiation-induced bystander effect. So these actually data also suggest that in the conditional medium, we have something that can cause bystander effect in a naive or unexposed animal. So what could these factors be? Could it be DNA? Could be RNA? Could be protein? Or it could sometimes people have proposed that the bystander effect factor are gas. Okay? So we start actually with the simple solution is we treat this conditional medium with either DNAs to destroy DNA, RNAs, or protease. As you can see here, that DNAs or RNAs treatment doesn't really affect the bystander response in the medium. But protease treatment completely abolish the bystander response. This is actually germ cell inhibition assays. So that actually makes things a bit easier for us because we can easily use the proteomic approach to find out what proteins are in the conditional mediums. That's what we did. We concentrated the conditional medium by about 100-fold, and then we run them with the control conditional medium, which means without the UV radiation, on the polyacrylamide gels, do the silver staining, and we can see there are 10 unique bands present in the UV conditional medium. Then we cut this band out, do the proteomic analysis. This was done with, in collaboration with Dr. Yi Zhaosheng at Chang'an University in Taiwan. So these 10 unique bands gave us about 19 proteins candidates. And so the question is, which protein or which proteins are the candidates? And that's when the reverse genetic in C. elegans actually kick in, really help us, okay? So what we did is we start doing the same assay, medium transfer assays in C. elegans, but doing in the RNAi background. So basically in C. elegans, we can do the RNAi by feeding the animal with RNA, double-stranded RNA expressing bacteria, okay? So we did that with all 19 candidate genes, and then we do the same assay, the germ cell death inhibition assays. And as you can see here, that this is actually controlled. One gene actually stands out. So when we do RNAi on this gene called CPR4, we pretty much abolish the bystander effect in the medium. And this actually turned out to be a gene encode a human homolog, one homolog of human cathepsin B protease. So to confirm this CPR4 gene is really important for the bystander effect that we're seeing in the conditional medium. We knock out the gene, again, by reverse genetics, and we can see that indeed the bystander effect disappears in the mediums prepared for conditional medium, prepared from the knockout animals. And we can put back a single copy CPR4 transgenes with the fragment, and we can completely restore the bystander effect. And we see the same thing in the conditioned medium prepared from the gamma irradiation. So that means that two different irradiating sources produce the same bystander effect in the medium. Okay. So what's CPR4 or what's cathepsin B? So it's a very interesting protease, cysteine protease. It's a dual function protease. It normally actually resides in the lysosome at a low pH environment. Is an exopeptidase. Okay. However, this protease has been shown to release out of the cell into extracellular space. And in this case, in the neutral pH condition, it instantly switched to become an endoprotease to cleave the proteins. Okay? And this protein has been shown to express constitutively in many different cell types, and interestingly, is a known cancer biomarker. So as I mentioned, this protein normally stays in the lysosome and only under special situations get released out of the cell. So could this radiation stimulation actually is the reason for this protein to be released into the out of the cell? And the result is yes. So we use this single copy transgene in the knockout mutant background because it has a flat tag, so we can detect the protein. And you can see that with UV irradiation, the conditional medium has this protein. Okay, but without UV irradiation, this UV control, we don't see the secretion of this protease. Because this protein is flat tag, we can actually immunodeplete this protein from the medium and then see whether the depleted medium actually still contains this bystander effect. And as you can see here, the anti-flat depleted medium, conditioned medium, lost any bystander effect. So this actually confirms that CPR4 protein actually is present in the conditional medium and is the major component that exert the bystander effect. So now the big question is, how is this protein get released out of the cell in response to radiation? And then how do unexposed cells or animals actually respond to this bystander factor? Here we actually decide, because this medium transfer technique in C. elegans is very laborious, it takes a long time, so we could not possibly do a genetic screen on that. So we actually decided to take a candidate approach. So we basically look for potential candidate that might affect or regulate the release of this CPR4 or the response to this CPR4. So C. elegans P53 homolog is a tumor suppressor gene called SEP1. It's known to mediate DNA damage repair and respond to either IR or UV radiation. So we started tested whether P53 is involved. And the answer is yes. As you can see, that just like knockout CPR4 animal, CP1, the P53 knockout animal, also lose bystander response. And we can see by quantitative RT-PCR analysis that upon UV irradiation or gamma irradiations, that CPR4 transcription is significantly increased, and this increase in transcription is blunt by inactivating the P53 in C. elegans. And if we knock out P53 gene in C. elegans, we see a greatly reduced secretion of this CPR4 protein. So this suggests that P53 in C. elegans is involved in increasing the CPR4 transcription as well as its secretion in response to irradiation. So here is our model, is that when cell in C. elegans get irradiated, P53 is activated, and that somehow either directly or indirectly increase or activate the transcription of CPR4 gene, and that will lead to more synthesis of CPR4 protein, and then in combination with some unknown mechanism that also in response to the radiation cause the increased secretion of CPR4. So the other end of the question is how do cells, actually unexposed cells, respond to the bystander factor CPR4? As I show you, that CPR4 can inhibit germ cell death, can cause increased embryonic lethality, can increase germ cell proliferation, can lead to DNA damage in germ cells. It can induce stress response. So we are looking basically for a gene that can elicit all these very different kind of response. And there are not that many factors in C. elegans that have been known to involve in multiple developmental process or stress response process. One gene actually stands out. This gene is DAP2. It's a very famous gene because this gene is known in C. elegans. It's a homolog of insulin-like receptor. It's known in C. elegans to be involved in apoptosis, cell proliferation, metabolism, stress response, and lifespans. So we started testing whether DAP2 actually is involved in radiation-induced bystander response in C. elegans. So the answer is yes, it's involved. So in this case, basically what we did, we actually had purified CPR4 protein. So this actually serves as a gain of function, sort of like gain of function mutant for us, that we treated the animal with CPR4, purified CPR4 protein. As you can see here, that it can actually inhibit, just mimicking what we see in the conditional medium inhibit germ cell death. However, if we actually inactivate the DAP2 protein, we don't see any further reduction of germ cell death. So this suggests that DAP2 actually is involved in mediating the bystander response induced by CPR4. And we see the same result in the germ cell proliferation assay, is that in wild-type animal, when the animal treated with CPR4 protein, we see an increased cell proliferation, but in DAP2 loss of function mutant, we don't see it. So all these results kind of suggesting that DAP2 actually potentially is acting downstream of CPR4 protein to mediate the bystander effect, at least these two different ones. So here we are connecting the dots that, as I just showed, that irradiation of the cell will trigger the secretion of CPR4 bystander factor through the P53 tumor suppressor genes, and then CPR4 protein either directly or indirectly add on the insulin receptor DAP2 to trigger multiple different bystander response. As I mentioned, that CPR4 is a homolog of human cathepsin B protease. It's a very interesting dual function protease. So we asked whether this protease activity is critical for bystander response. And the answer is yes. Again, here you can see that with purified CPR4 protein, wild-type protein can cause, when incubated with a naive wild-type animal, we can see a significant increase in embryonic lethality. But when we actually mutate the catalytic site of CPR4, this is actually a protease DAP version that we don't see any of this increased embryonic lethality. Very interestingly, that when we use human purified cathepsin B protease to treat the wild-type naive C. elegans animal, we also see a very similar increase in embryonic lethality. So this actually suggests that maybe this bystander response pathway could be conserved. And indeed, when we actually quickly look at whether the human cell line, different cell line can respond to radiation to increase the expression of the protein, you can see that in all cases, we can see increased synthesis of the cathepsin B protease in response to either UV or X-ray irradiation. Of course, one question is because this mediate transfer method actually deals with the bystander response between the animal. Okay, so it's an inter-animal bystander response. And so does this really relate to what we're seeing in human radiotherapy? That usually refers to the side effects on unexposed tissue in the same treated individual. So we actually need to come up with a similar localized irradiation model to test whether the bystander factor we identify is involved in a bystander response similar to the human radiotherapy. So we did that. So in this case, we basically used UV. We used the confocal microscope to focus the UV beams on only the head region of the C. elegans animals. And then we asked whether in any of this unexposed area, for example, unexposed oocyte, unexposed germline, or posterior area of the animal actually had any bystander response. The first one we tested is the localized irradiation in the head. Then we look at the germ cells. Okay, so in this case, we use a Hus1 GFP, which is a DNA damage checkpoint protein similar to the yeast Hus1 protein. This protein actually is known to form a discrete foci on double-stranded DNA break. Okay, as you can see here, that after we have localized UV irradiation in the head, the non-irradiated control doesn't have any Hus1 GFP foci. But with localized irradiated animal, we see multiple discrete foci of Hus1 GFP, suggesting that even the targeted irradiation in the head can induce DNA damage in the unexposed germ cell. And this DNA damage induced by localized irradiation is dependent on CPR4, because when we knock out the genes, that we don't see any of this DNA damage. Another assay, that stress response is one common symptom seen in radiotherapies. So we actually, here again, we are using another reporter gene, which is a He-short protein 4 in C. elegans, which is known to induce stress response in response to all sorts of stress. In this case, it's irradiation. As you can see here, that localized irradiation in the head, in the control, we don't see any GFP, but in a localized irradiated animal, we see an increased response in the posterior area, which did not receive any irradiation. Again, this stress response is dependent on both CPR4 and DAP2. If we mutate either genes, we abolish this bystander response. So finally, as I mentioned, that sometimes that this radiotherapy can induce even secondary tumor. And we test this in C. elegans using a C. elegans tumor model. So in this case, we are using a germline C. elegans tumor, which actually is a triple mutant, 3 genes are mutated. And this is a very interesting tumor model because it's temperature sensitive. At permissive or semi-permissive temperature, only half of the animal has tumor, okay? And what we did is we actually hit the animal again locally at the head with UV beam, and then we actually examine whether this germline tumor actually is reduced or increased. So in this case, what we're seeing is that when we hit it at the head, then the unexposed germline actually has increased tumor frequency as well as increased tumor size. So this suggests that even this radiation-induced side effect can induce tumor in a sensitized genetic background. And we can completely block this with using a cathepsin B inhibitor that we just identified. I'll talk about it called RIBI1. As you can see that when we treat the animal with RIBI1 first, and then hit at the head with UV beam, we don't see any increased tumor or increased tumor site. So what next? We're doing a lot of things right now. We're trying to, because cathepsin B is a protease, we're trying to identify what are the substrate of CPR4 or cathepsin B that actually play a role in bystander effect. we are trying to figure out the mechanism and signaling pathway that involved in mediating very different CPR4 bystander response. For example, genome instability, stress response, cell proliferation, and apoptosis. We're trying to figure out in the process of testing whether cathepsin B actually mediate the similar bystander response in mammalian system, and also we are trying to test whether the same signaling pathway, the same bystander factor, for example, cathepsin B is involved in other stress response, for example, chemotherapy or other stress response. So one reason actually we are using C. elegans as an animal model to study bystander effect, which actually originally had nothing to do with us, is because we are interested in helping treating human disease. So we actually started to do the drug screen in C. elegans to identify compound that can alleviate side effects caused by radiotherapies. So why actually doing this in C. elegans? There are multiple really unique advantages. One is C. elegans is really going fast, only 2.5 days per generations. And this is actually a major reason now in the funding situation is so tight that you can find a way to do a cheap drug screen. That's always good. And C. elegans actually is cheap. It costs no more than growing bacteria. Okay. But the more important reason about doing drug screening in C. elegans is that the screen actually are conducted in the whole animal under the normal cellular context. And this is especially true for this radiation-induced bystander effect because it involves long-range cell signaling, cell-cell interaction. So you can possibly do drug screen on this kind of bystander effect in vitro system. Of course, C. elegans, as I just mentioned, has really powerful genetic and reverse genetic tool that can facilitate the target ID, which have been the bottleneck of drug discovery. So over the last 10 years or 20 years, last 10 years, we have been doing a lot of drug screen first on the Hep B virus and on some other disease model we have actually established a really nice system to do drug screening in C. elegans. So in the past, only limited chemical screen can be done in C. elegans on water-soluble compounds. Now we actually had ways, for example, involve oil solvent that can do drug screen with hydrophobic compound, because a lot of drugs actually are hydrophobic, okay? To make the long story short, all right, so the current therapy, the current solution for treating side effect resulting from radiotherapies are quite limited because, again, here they are really big. Radio-oncology department this should be quite familiar. There are currently a few different solutions, for example, Exile to treat the dry mouth in head and neck cancer, sterile to treat swelling in body tissues, some topical agents or growth factor. They are all targeted to specific radiation-induced side effect, but they cannot generally treat the side effect, and they actually usually don't improve cancer killing when combined with radiotherapy. So we are aiming to identify compounds that can generally treat radiation-induced side effect and also improve cancer killing. To make a long story short, we actually have multiple compounds, and two looks really promising right now from the C. elegans drug screen. They are isolated as inhibitor of the cathepsin B protease. And they can, in the C. elegans model, they can inhibit multiple radiation-induced side effects, including chromosome damage, stress response, increased embryonic vitality. One of the compounds we call RIB1, RIB means RIB inhibitor, can even actually enhance cancer killing by radiotherapy in the C. elegans tumor model. So this is just a quick data slide that RIB1 can potently inhibit the cathepsin B, both the C. elegans cathepsin B protease and the human cathepsin B protease. And this compound in the C. elegans model does not show any toxicity because it doesn't affect C. elegans embryonic vitality, larval arrest, or brood size, which are three critical features about testing the C. elegans drug in C. elegans. And as I said, that this compound, both RIB1 and RIB2, can potently inhibit all the radiation-induced side effects that we tested. And here, example, you can see RIB1 and 2 inhibit germ cell damage, embryonic lethality, and stress response. Most interestingly, when combined with radiotherapy, RIB1 can even improve killing of the cancer. So let me walk you through this slide. So for example, 20-grade X-ray irradiation of this the C. elegans germline tumor model I just mentioned doesn't really do anything to reduce the tumor frequency or the tumor size. But when in combination with our RIB1 compound, it can reduce the tumor frequency and shrink the size of the tumor. So we are really excited about this. So as I mentioned, that the current solution for treating side effects in radiotherapy are quite limited. They are used normally target to a specific side effect. So our compound could be the new drug for treating multiple side effects induced by radiotherapy, but at the same time can improve the cancer cell killing. And so the big questions, and that's part of the reason I really I want to come to this Angel campus is look for collaborator can work with us to test whether RIB1 can work to enhance cancer killing and prevent side effects in the mouse and human model. Lastly, so far I've been mentioning that these side effects seen in the radiotherapy, radiation are all bad, all adverse, right? But as you can see, this is a question that actually intrigued the field for many, many years. So can this radiation-induced side effect be beneficial, could be protected rather than destructive? So we actually decided to test that in one, and C. elegans actually is a great model to test this. Is that, for example, we want to test could repeat the side effect can help organism to adapt to changing environment. For example, when in the wild, the animal move from a place to a place has high UV radiation or gamma irradiation. So the way we do this, and C. elegans is pretty much the best organism to do this because it's short life cycle time, okay? It's only 2.5 days. So what we did is we actually irradiate the C. elegans animal, water animal with a low dose, 100 joules, okay, for 2 generations. And then we leave them alone for 2 more generations to let them grow and have progenies. And then in the 5th generation, we start hitting them with high UV dose, okay? That's a triple high UV dose. And as expected, we see many, and so to do this, we have to clone out hundreds of animals, sometimes thousands of animals. And to do this, and as expected, you can see most of the animals are dead, okay? But we get some escaper that actually has many progeny that can survive. And when we analyze this, and we got very interesting result, okay? So this N2 is C. elegans water animal. You can see that without UV radiation, you get some very low percentage, about 3% escape, okay? But with the low dose, 2 pre-exposed low dose UV radiation, we see a significant increase in survival, okay? And that kind of suggests it's an adapted UV resistant response, okay? So we can see about 12% of the water animal now acquire after 2 low-dose UV radiation acquire the UV resistance, okay? However, if we do this in the CPR4 loss of function animal, so this adaptive UV resistance is abolished, okay? So now we are looking at do RIB actually radiation-induced bystander effect actually has other beneficial effects. So I'm going to close the talk by thanking the people who actually did all the work. So Yipan, Zaman, and Linjin and Han Zheng are 4 really courageous graduate students actually who took on the project, because at the beginning when I tried to ask students and postdocs to do the project, they refuse because they think it's too risky. And Liang Qian and Tian Kan and Bing Yu are postdoc and graduate students who also assist in this study. And this actually was done in collaboration with Shohei Mitani in Tokyo Women's Medical University and Yi Zhaosong's lab in Chang'an University. So I'll stop here for questions. Thank you. Yes, Jens.
First of all, I think that last bit on adaptation actually really validates what Nancy said about what doesn't kill you makes you stronger.
Yes.
So with that, it looked like you've gone several generations after the UV exposure.
Up to 2 low-dose UV radiation.
Is that are you selecting out stable genetic, resistance?
So
could it still be a temporary mechanism?
No, it's not temporary. All this actually stable actually became UV resistant. So what happened is, as I mentioned, this bystander response involved DNA damage. Okay, that might create mutations. Okay, and these mutations could somehow create UV resistant mutant.
So you think it's just basically allowing greater vulnerability by allowing cells to have mutations to persist?
Yes.
So if you did a general assay for just alterations and mutations, you would actually see that it was basically facilitating just a higher mutation rate.
Yes. And this, as I mentioned, is very interesting. It's CPR4 dependent. If we knock out CPR4, we don't see that. So that means that bystander effect is really playing a role in this process, adaptive evolution. Yes?
So early on, I think you showed a lot of the graph with UV radiation.
Mm-hmm.
If you looked at X-ray radiation or gamma radiation and see the same effects, in what doses, is there a threshold, for example, where CPR4 is induced, and have you looked at what kind of dose-dependent responses?
Yes, we did. So basically, all the expansion, because time limit, I did not show all the data on gamma irradiation. We basically, in our Nature paper, we basically do everything side by side. UV, gamma irradiation. And so gamma irradiation, I remember, is, I can't remember the exact number. It's probably like a 200 gray or something like that. Yeah. Silicon actually is quite resistant to irradiation. So, yes.
Another question. So, presumably when you go into minus for patients, you could have different cells that will secrete different levels, even though it's the same dose. Have you looked at developing a screen where you could look at, for example, the blood and CPR4 levels after radiation and see how they differ in population?
No, we have not, because my lab is mainly work on C. elegans, so we don't deal with patients, we don't deal with animals. So the only animal model we work on right now is Hep B virus, okay? So that's part of the reason I'm coming to, and radio-induced side effect and radio-oncology. Yes?
Mechanistically, direct damage to a cell by radiation and the bystander effect are going to be quite different.
Yes.
So you expect your RIBI to inhibit the latter. So do you have any idea from your C. elegans model which of these contributes more to tissue damage, or can you kind of?
I think direct damage actually contribute much more to the tissue get irradiated. We actually have data on that. For example, embryonic lethality. If we just directly irradiate the embryo, okay, or they are pretty much 80 or 90% dead, okay? But for radiation-induced side effects, much less. We only see 5 or 6% in the unexposed oocyte that has lethality. Yeah. No, we actually want to prevent the side effect on the unexposed cell.
Right,
That's
mediated by CPR, which is
that's somehow affected by CPR. Yeah.
Yes, and following up on that question, so vaccine B in human cancers is
most likely upregulated?
Yes, that's known. So cathepsin B actually is known to upregulate it, and that's why I have a slide showing that it's a known cancer biomarker. So, Xiaojin?
For the potential CPR4 protein substrates,
Yes.
now if they are in general ubiquitinated or segregated, so you can narrow down which group.
That's a very good question. So we are trying to, before we actually send the paper out, I told my student we had to figure out a substrate, otherwise we are going to get killed. And we tried for 1 year, we couldn't find a substrate, but now we have very good candidate right now. It's not ubiquitinated, yeah.
David's been down there with this hand.
David.
Whether known ligands of, is it DAF2, the insulin-like receptor, affect the bystander effect?
That's a very good question. So we are testing that. So the problem with DAP2 is C. elegans actually has like 30-something, 37, yeah, I remember correctly the number. That's insulin-like ligand. So we are doing individually right now. So that's a really good question, but we don't know, yeah. Because the number is insulin-like ligand is just too big. And we also had to deal with potential redundancy. So it may, even one of the insulin ligand is involved, it may not be enough. You have to knock out multiple. And that's a daunting task because how can we deal with 37? Yeah.
What's the mechanism by which CPR4 is being released?
That's a great question. I don't know. The part of the reason I already showed CPR4 gene transcription is increased, but I don't think that's sufficient. It must couple with something else, possibly the secretion machinery. So we are testing some of the candidates right now, whether they are altered in response to the bystander, the radiation.
Does it localize
Kelly.
to the target cells? Does it localize to the cells where you're seeing the bystander effects, or is it something that's happening in circulation?
I think it's something happening in circulation. We have pretty good evidence because C. elegans actually has an artificial circulation system called pseudocyte, pseudosorum, sorry. And that actually can circulate secretory factor, and CPR4 is a secretory factor. Yeah. All right, thank you.